Good morning, everybody. Welcome on a sunny and cold Tuesday morning at Chem 11:05 with your host, me, Dr. White. Hi, everybody. Let me remind you, next Monday, I will be giving test number 4 after lecture. Also, next Monday is the deadline for the extra credit project if you're going to do it. If you're going to do it, I'd highly recommend email me your project before you upload it to Blackboard, and I'll look at it. If anything's wrong, I'll let you know so you can fix it so you can get 10, yep, 10 big ones. All right, today's session, first of all, we're going to do the lab, and then I'll continue with organic chemistry. That's not what I want. That's what I want. Nope. Hold on. That's what I want. All right, let's get going. Let me make sure you can see what I have on my screen. And you can. All right, today's lab deals with the determination of acetic acid. What you're going to be doing is finding the concentration of acetic acid in vinegar using a titration. Now, let me remind you, you'll be working with vinegar and 1 molar sodium hydroxide. You should wear goggles, you should wear gloves, and an apron would be a good thing too. I'd highly recommend if you have a marble top, like I've talked about in the past, use a tray. You can get those cheap aluminum trays for $2 or $3 at Walmart and other places. All right, now we've talked about acid and base and pH. Well, we're not going to be using pH in this lab, but what we will be doing is the titration. And here, the reaction is acetic acid plus sodium hydroxide gives you sodium acetate plus water. And let's go back and do a little review. Here you have an acid, a base, you have a proton transfer, and you make a salt. Now, in our case, you'll have acetic acid, and this is a better way of writing it. And here's your acidic proton. And the base will be sodium hydroxide. And you will react the two together, and you'll have proton transfer. You'll form this salt plus water. Now, when we talk about, I guess we should talk about pH. Here's pH scale, goes from 0 to 14, neutral is 7. Below 7 is acidic. As you add base to the acetic acid, which is the vinegar solution, the pH is going to go up. And when it hits 7, all the acid will react with all the base. It will be neutralized. That's why they call 7 neutral. Well, you're going to be using a pH indicator. And that will be phenolphthalein, the most widely used pH indicator. Below here, this is a clear, when your pH indicator is below 7, it's clear. As soon as you get one molecule above at 7 and one molecule above, it turns a pink color, or I like to think of it as a wine color. And you'll be able to determine how much sodium hydroxide is added using a syringe to neutralize all the acetic acid and vinegar. That's the titration. Now, I didn't have it on this machine, so time to go looking. Hold on, I found it earlier. Here we go.
When you and the team are pitching a new product, Grammarly's tone suggestions?
All right, now here you'll be using an Erlenmeyer like this. You'll have a known amount of acetic acid and water in there. Now they're using a burette. You don't have one at home, you'll use a syringe. And as you add the base, which they're adding very quickly right now, and you once in a while, they're showing you how to swirl the Erlenmeyer. I'm going to skip forward. Notice how it turns that pink or wine color. That's the indicator. But when you swirl it, you're causing the molecules to react, and that color goes away. Now, you keep on adding base until the lightest color stays that. You don't want to go too far. That's called overshooting your endpoint. You don't want that. No, you don't. So, see how it's turning that color? And you'll add a little at a time, swirl it, and notice how when she swirls it, it goes away because the base and acid are still reacting. Now, as you get closer to that color, slow down, add a little at a time, swirl, and then add some more. And if you have a piece of white paper underneath, you can see the color change even easier. And now if you notice, the color does not go away. And that's your endpoint. Now, if you were to add more, and notice the color stays that very light, and that's your endpoint. Now, if you look at, and I'm in student view so you can see what you would see, you'll see I have the lab instructions, the lab report, and at the bottom, I have note, just do activity 1 and 2 for this lab. Don't do 3. And See today's video for further guidance on activity 2, and be sure to answer the question on page 4. Now, let's look at today's lab. What are you going to be doing? You're going to be having a lot of fun. That's what you're going to be doing. Now, you'll need this equipment right here. And you should have also a bottle of vinegar in the kit. You'll also use the phenolphthalein indicator and the sodium hydroxide. Notice it's 1 m. That means it's 1 molar. And as they say here, use your goggles, gloves, and apron. And if you get anything on you, rinse it with cold water for about 15 minutes. You shouldn't be eating or chewing gum, and wash your hands after you're done with soap and water. All the waste from today's lab can be safely poured down your sink and let the water run for about 5 minutes, cold water, after you do that. Now, everything you'll need for activity 1 is in the kit. For activity 2, you'll need everything, but you will have to supply a vinegar sample. Use something different than the vinegar brand that's in your kit. In other words, use one that you have in your house. And I think you all have vinegar in your house. If not, go knock on your neighbor's door and borrow some. All right. Now, in part 1, here are the steps. You'll take your 10 milliliter graduate, put it on the balance with an eyedropper from the bottle of vinegar, put in about 5 milliliters of vinegar. You can look on the graduate. It doesn't have to be 5.0, but try and get it close to it. At that point, remember when you put the graduated cylinder, make sure you tear the balance so it's at 0 before you put the vinegar in there. After you get to about 5, record the mass of the vinegar in the graduated cylinder. Pour that carefully into the 25 Erlenmeyer flask. They say add 2 drops. You can add 3 or 4. And then swirl it like this. Then add approximately 10 milliliters of the 1 molar sodium hydroxide to a small plastic cup. One of these, you should have some left over. And what you'll do is with the syringe, you'll fill it to almost all the way. And then record the volume of the syringe to 1/10 of a milliliter in your data. Then what you'll do is place the flask containing the vinegar on a sheet of white paper and slowly add small amounts of the sodium hydroxide. If you don't have the phenolphthalein in there, it won't work. And when I've done this lab with a burette, students will come to me, mine isn't changing. Did you put phenolphthalein in there? No. And that's why you need to put up here in step 6, 2 or 3 or 4 drops are better of the phenolphthalein solution in there, or it won't change color. Now, when you start seeing the color persist but still go away when you swirl it, start adding the NaOH, sodium hydroxide, dropwise, slowly. Now, they give a good thing right here to gain more control, to get single drops. Don't push it from the top. Hold it from near the barrel, and that will help control it. And then as you're going slowly, add a little swirl. When it stays the lightest pink, you don't want it to get dark pink because you overshot your endpoint. Endpoint is when it first changes color and stays that color. Very light, but you can see it. Now, at this point, you'll record the volume on the syringe as you've measured. And the difference between record the final volume and the initial volume is the total volume. Now, the titrated vinegar down the sink, put on some cold water, rinse it a couple times with water, and then you'll repeat steps 1 through 16 again, do a second titration. And then you'll do it a third time. In real life, in a lab, when we're doing titrations to measure something, you always do it minimum duplicate twice. Now, let's go through to part 2, activity 2, then I'll go through the calculations. All right, now you'll do activity 2. You'll take the vinegar solution or sample, not the one with a kit, but something you have at home. And it can be cider, malt vinegar, balsamic vinegar, even rice wine vinegar. They don't have it here. By the way, I have cider, balsamic, and rice wine. I don't have malt. I've never had that. I don't know. I just never have. And what you'll do is for activity 2, you'll do 3 times the activity 1 steps 1 through 17. In other words, do 16, 1 through 16, 3 times with the other vinegar. And you'll put the data in this table too. Now, if you notice, you have to do some calculations. The volume delivered is final minus initial. And the mass of the vinegar is what you weighed out. Now, things like the volume of the vinegar, moles of NaOH delivered, moles of acetic acid in the sample, mass of acetic acid, molarity, that you have to calculate. And how do you do that? Well, good news. If you look at lab 12, you'll see right above where it says note, says lab 12 calculations. Don't tell anybody I was nice and helped you out. Shh. Don't tell anybody. Remember, I don't want to ruin my image here. I want people to think I'm mean and nasty. All right, let's look at this lab calculation. That's not it. All right, now you'll see I did it in handwriting. It was just quicker. Milliliters of vinegar. How do you calculate that? You have the mass of the vinegar in grams, and this should be 1 milliliter is 1.005 grams. That should be a 1 up there, not a G. Or, okay, and that will give you Z milliliters of vinegar. Now, for the moles of sodium hydroxide used, H is the milliliters you use that you calculated. And 1 mole, that's the molarity per 1 molar NaOH is 1 mole of NaOH per 1,000 milliliters. This will give you the moles G of NaOH. Now, how many moles of acetic acid? Well, hopefully you learned this from test 3. We look at the balanced chemical equation again. And this is the balanced chemical equation. Remember, the coefficients give you molar quantities. And when there's no coefficient, it's 1. And this tells you, I think I'm going to sneeze, 1 mole of acetic acid reacts with 1 mole of sodium hydroxide. So if we go back to our calculations, to get the moles of acetic acid in the vinegar, you have gram of G from this step multiplied times 1 mole of acetic acid per 1 mole of NaOH. G is the same as I. Now, to get the molarity of the vinegar or the acetic acid in the vinegar, I moles of acetic acid times 1,000 milliliters divided by Z milliliters of vinegar. And that gives you your molarity of acetic acid. Where do you get this number Z? Way up here. Remember, that G should be a 1. My mistake. I'll try and correct it later today and repost this. And that's how you do your calculations. Now, here they ask you to do the calculated molarity and then the percent mass. Ignore that and do the average calculated molarity, not the mass. And if you notice table 1, you'll do these calculations. You'll do the same thing in table 2. You'll get a mass of vinegar. You can calculate the volume like I showed you. Initial, final volume, you measure. Volume delivered is final minus initial volume. I show you how to do the moles of NaOH in that calculation, show you how to do the acetic acid moles, and also the molarity. Mass of acetic acid, you don't really have to do. Same thing with percent mass, but calculate not percent mass, but percent average molarity. That's not what I wanted. All right, sorry about the technical delay there. If you look at the lab report, you have to fill in these tables, which I showed you how to do for 1 and 2. Remember, we're not doing activity 3. Remember on page 4, and I have it on the lab in Blackboard, answer question 1 on page 4. And that's today's lab. Now, remember, wash your hands afterward, wear protection, goggles and gloves and an apron. All the liquids when you're done that you titrated, just go down your sink. Afterward, let the cold water run for about 5 minutes, I would. And don't forget to wash your hands when you're done. And that's today's lab. Don't forget, last week's lab is due today. And with that, if you have any questions about any lab, especially this one, always feel free to come to my office hour. I have one tonight from 6 to 7:15 and tomorrow night. And a quick reminder, next Monday is test 4. All right, let me close a few things. All right, let's take a look and get back to organic chemistry. This will be on test 4 coming up next Monday. And when we talk about organic chemistry, the beauty of it is, and it truly is, as I mentioned yesterday, there are key functional groups that you find in molecules, organic molecules, like the ones here. And the good thing about functional groups is the fact that their chemistry, a reaction of certain functional group will yield the same type of product most of the time, about 98%. There are exceptions. That's when someone like me, a PhD organic chemist, comes in to get around those exceptions. So what that means is when you have a functional group and you react it to certain reagents, no matter what kind of molecule it is, you'll get the same kind of new product with a new functional group in it. And this is so strong, so powerful. Now let's look at some key reactions. And these are like general reactions that you can apply to specific cases. The first one is combustion. You take a saturated hydrocarbon, react with oxygen, and you need a spark or heat source. You burn it, called combustion. You'll always get carbon dioxide, water, and heat, which is not a chemical, but it's an important part of the reaction. Now, organic chemists are lazy. We are. And if we don't have to balance an equation because we don't need to, we don't. And when we talk about reactions, we don't balance equations unless you have to find out charge weights. What I mean by charge weights is how much of each chemical do you have to use to make a certain one, a product. If you're not doing that, we don't balance the equation. We're lazy. So let's look at combustion reaction. And how do you use this general reaction in organic chemistry? And by the way, I better be subtle. You know something, I might even make it 4 points, 3 or 4 points each. And the question is, give the product for, and you see, A, this is propane. This is oxygen. What do you get? Well, to make it easier, I'll tell you get 2 products. That's why I'll put a plus sign there. I don't have to, but I will. Well, how do you do this? Well, you know functional group chemistry, the general reactions. And what is propane? It's a saturated hydrocarbon. And oxygen is oxygen. And you know the general reaction always happens saturated hydrocarbon oxygen, you will get carbon dioxide plus water. We don't put the heat down because that's not a chemical for this course. So if I have propane, it's a saturated hydrocarbon reacting with oxygen, you know what I'm going to get? Carbon dioxide and water. And that's how you use the general reaction to predict specific products of a specific reaction. Ooh, this was fun. Let's do another one. And I'll let you try this one first. What would be the products of that reaction? Remember, you're trying to figure out what are the products of that reaction. And for you, those of you watching the video, if you need more time, hit the pause button and then start again when you're ready to go. All right, this is butane. And that's the liquid in those lighters. And it's a saturated hydrocarbon. What's a saturated hydrocarbon? A molecule with only carbon and hydrogen atoms and only carbon-carbon single bonds, which this does. And what you're reacting it with oxygen, and what do you always get? Carbon dioxide plus water. So what's the products going to be for butane? Carbon dioxide and water. And that's the strength. And I'll show you some more examples. All right, just a quick. If you look right here on your screen, let me make sure you're seeing it. Yep. Right here, they have custom solid colored standard flint cigarette lighter. Not you should be smoking cigarettes. Luckily, I gave that decades ago or I'd be dead now. But the liquid inside here, let's see if they have one where you can see it. Like here, there's a liquid in there that's butane. And when you click it, you're lighting a spark that causes the oxygen to react with the butane. You see the flame, that's the heat, but you also get carbon dioxide and water. Oh, let's do one more. 3 points, and you should know how to do this. And the beauty of functional group, oh, by the way, you can bend arrows, I do, organic chemists, is you don't have to memorize each example. What do you get? Here, you look at this molecule, you're reacting with oxygen. What are the products? And I'll help you out. The plus tells you 2 products that you get in that chemical reaction. Your turn. Again, your job right now, 3 points each, give the product or products for that reaction. If you want, hit the pause button if you're watching this video. And I'll continue. I assume you gave yourself enough time to figure it out. Now, this is octane. Actually N-octane. And actually not N, you don't have to have the N there. It's octane. It's a major component in gasoline. And in your car engine, the engine burns octane combustion in this reaction. What's octane? A saturated hydrocarbon. It's reacting with oxygen, and you've memorized this reaction. In organic chemistry, there are a lot of reactions you have to memorize. When I teach Chem 1212, which is a one semester organic, they have about 55 of these to memorize. I think I know about 500 of them, but I have a PhD in organic chemistry. And I'm a synthetic organic chemist, which means I know a lot of these. Well, you learned saturated hydrocarbon oxygen will give carbon dioxide and water. So if I take octane, a saturated hydrocarbon react with oxygen, what do I get? Carbon dioxide and water. Now, if you've ever been behind a car in the winter, a real cold day, you'll see some of the cars out of their tailpipe is a liquid coming out, and you'll say, oh no, it's leaking gasoline, it's going to blow up. No, that's not gasoline. What happened was their muffler and tailpipe are still very cold. The reaction in their car engine gives off water as a gas. But the cold muffler and tailpipe condense that gas form, called a vapor, of water to a liquid. And that's what you're seeing. Carbon dioxide is not cold enough to ever condense that to a liquid. And anyways, well, I'm not going to get into that. So you should know how to do this. How do you do it? You'll learn for this class, I'm only asking you to learn two general reactions. This is one of them. Saturated hydrocarbon oxygen gives CO2, carbon dioxide, and water. And you've just seen how we apply it. Now, on a test, do you have to write this underneath? When I teach organic chemistry, some of my top students do that for every reaction like this. When I teach Chem 12, one semester organic chemistry, and I've done that a number of years here, some of my best students don't, but it's in their brain. Either way, you need to know it. All right, let's move on to another reaction. Next, I'd like to talk about hydrogenation. I'll never ask on a test what is hydrogenation, just like I'll never ask what's combustion, but you need to know how to apply it. Well, hydrogenation is the addition of hydrogen gas, H2, to an alkene, double carbon-carbon double bond, or an alkyne, a triple bond. I should have had here. And you need a catalyst like platinum, palladium, or nickel. And here I just have any double bond. I'll show this better on my whiteboard. You react with hydrogen, a catalyst, and you break one of the two bonds. Those two lines are two bonds, called a pi bond you're breaking, and each carbon of double bond gets a hydrogen. And I'm being subtle again. And know this. All right, what should you know? Now, organic chemists, I'm just going to show are lazy. So anytime you have a double bond, I won't do it for a triple bond, and you add hydrogen and a catalyst. And the catalyst can be nickel, platinum, or palladium. and this has one pi bond. I'll never ask you one sigma bond. And you break the pi bond, and each carbon of the double bond gets a hydrogen. This is hydrogenation. Later this chapter, I'll talk about this is what they use to make hydrogenated fats and oils that, even though unhealthy, are in many things you use. And like margarine used to have this. I don't know if they've changed from that. But one thing I should mention, I can write a chemical reaction in organic chemistry like this. A plus B makes C, or organic chemists will do it like this also, which is what I've done here. All right, let's take this for a spin. Now, if we look at this, except for combustion, you don't break carbon-carbon single bonds. A double bond, yes. A single bond, no. Now, if we look at this molecule, what's different? What's not a carbon-carbon single bond or carbon-hydrogen atom? A double bond. So we have the only thing important that would react in here is a double bond reacting with hydrogen gas. And what is nickel? Ni, a catalyst. And the catalyst is there something that makes the reaction go faster. If you didn't have a catalyst here, you could wait a couple months or more, nothing would happen. With a catalyst and the right temperature, this is done in 4 hours or less, which is quite good. And the catalyst can be nickel, platinum, or palladium. And you break the pi bond. Notice here I have 2 bonds. Now I only have 1 because I broke it. And each carbon of that double bond, these 2 carbons, get this. Now, you don't break carbon-carbon single bonds. So I start with 3, I'll end up with 3. But notice I broke one of the pi bonds, and each carbon will get a hydrogen. Now, since there are 4 bonds to carbon, you can figure out this will now have 3, this will have 1 more, this will have 2. And that would be the product. And you need to know how to do that, 3 points. Let's do another one. And the question is, give the organic product or products that are following. 3 points each. Your turn. I'll hit pause if you need more time. And let's go. What's different in this molecule? What's not a carbon-carbon single bond or carbon-hydrogen atom? A double bond. And that's where all the fun's going to happen. That's where the action is. And what are we reacting with? Hydrogen gas. And what's platinum? It's a catalyst. And the catalyst can be nickel, platinum, or palladium. And you break the pi bond. There's 1 pi bond. That's what it's called. If you were taking my Chem 1212, I teach you more about pi and sigma bonds. And each carbon of the double bond gets a hydrogen. You break carbon-carbon single bonds? No. So you have 4 carbons. Notice I broke the pi bond right here. And these 2 carbons of the double bond, these 2, each get a hydrogen. And there are 4 bonds to carbon, so I can count how many hydrogens more I need. And that's how you do it. If I look at the clock, it's time for a 5-minute break. I'm going to take a 5-minute break. I can get up and stretch, and we'll continue in 5 minutes. let's get back to work. All right, we're talking about general reactions. I just went through hydrogenation. Now, this little thing, my first research project in the chemical industry when I was a bench chemist, research chemist, was hydrogenation. Now, I had a triple bond between carbon and nitrogen that I was working with. And an interesting thing I should teach you right now, if you see on your screen here, this is called a tank wagon. And in the chemical industry, these are standardized. Tank wagons are always 40,000 pounds, 5,000 gallons. And in the chemical industry, in a lab, you work with about a pound of chemical. And then you scale it up to what's called a pilot plant, where you work with a 1 drum, 55 gallons, 500 pounds. And then when you go in a chemical plant, at least the ones I've worked in, the smallest one to run a reaction, we call that a batch, is 40,000 pounds, 5,000 gallons, one of these. In one reaction, you do that. Oh, it's beautiful. It's fun. It's exciting. Now, in my career, the largest single reaction I ever did was at one company, and that was about 160,000 pounds, which would be 4 of those tank wagons of chemicals. And you don't want to make mistakes. One lesson I learned very early on is you never experiment in a plant. Because if you're dealing with 40,000 pounds, that's about $40,000. And if you mess up, then you've wasted $40,000. And you don't stay employed that long when you do that. Never happened to me. But that's the fun part of industrial chemistry for me. All right, let's move on. Now, I'm going to talk about some organic chemistry in your daily life. Some of this, click, the switch will be off. And some of these are areas I've worked in the chemical industry. Now, the first one, let's talk about fats and oils. Now, fats and oils are called a triglyceride, also known as a triester. An ester is a functional group, and fats and oils contain 3 of them. Switches click off. Now, switches on, you should know. What's a fat? It's a triglyceride that's a solid or semi-solid at room temperature. An oil is a triglyceride that's a liquid at room temperature. And if you look at butter, is made up of mainly animal fat or from milk or solids. And it's a solid. Now, what's a semi-solid? Well, if you keep a stick of butter out on the table for a long time or in your kitchen outside the refrigerator, if you were to push on it, it's sort of solid, but your finger will go in. Now, you know what a liquid is. And if you look at vegetable oil, corn oil, olive oil, hold on, I got to answer this. Sorry about that. Important phone call, someone I was expecting. All right. So you should know a fat, remember, switches on, click, is a solid or semi-solid at room temperature. An oil is a triglyceride that is a liquid at room temperature. And when I went to one doctor first time a couple of years ago, I had my annual checkup or a first checkup. They did blood tests, and he called me back and said, You know, your triglycerides are way too high. If you don't bring them down and a few other things, you'll be dead in 3 to 5 years. This was 10 years ago. So I followed his directions and brought my triglycerides down by eating a lot less things with fat and oil in it. All right, now, switches off here, but I should tell you, I worked in this industry. There are different types of fats and oils. Beef fat is called tallow. Pig's fat is called yellow and white grease. There are other kinds of oils. Now, rapeseed oil is very heavily used throughout the world because it's very inexpensive. When they tried to sell it in the United States, housewives wouldn't buy it. Rapeseed oil? No. Well, it's made mainly in Canada, and someone smart there said, well, it's an oil. We make it in Canada. Let's call, instead of rapeseed, we'll call it canola oil. I think I'm spelling it right. And you can go into any supermarket now and buy canola oil, where everywhere else in the world, it's called rapeseed oil. Now, I work for a company that purchased tallow and white and yellow grease to make chemicals that are other companies we sold to make chemicals like your soap, your fabric softener, and a whole lot of other things. And we would buy tallow and yellow grease, white grease, to the tune of about 2 to 3 million pounds. Are you ready for this? Per week. And we used a lot of it. It was big time. And I was the laboratory manager. I was in charge of research for North America and their quality control here in Chicago plant. All right, now, switches off, but in your stomach, here's a reaction. When you consume fat or oil, you have acid, and the acid H plus is hydrochloric acid. And you have water, and it breaks it down to fatty acids and glycerin. Those are molecules that your body uses. Now, when we talk about fats and oils and everything, this is something you should know. And how does soap work? Now, soap works with what's called surface active agents, which is shortened to surfactants. You know it as soap. Now, you should know what is soap. Now, soap, and you should know how to draw this. Let me get the right color ink. Soap has what's called a nonpolar tail. And a polar head. And you learned about like dissolves like. And what is important is dirt and grease, things you're trying to clean off things, are nonpolar. Water is polar. So if you have grease or dirt on your hands, just take water, it's still going to be there. How do you get it off? Well, you use, and the most important piece of this puzzle is, I'm going a real large font because it's so important. Use like dissolves like. Things that are polar are soluble in polar things. Things that are nonpolar are soluble in nonpolar things. The two you can't mix together. So how do you do this? Well, let's take a look at a little piece of dirt on your hand. You use now soap and water, and the soap molecules, the nonpolar tail looks at dirt, it knows like dissolves like, and says, you're nonpolar, I'm nonpolar, I'm going to be attracted to you. And it is. And the polar head wants to stay way away from the dirt because it's polar and dirt is nonpolar. Well, you form a lot of these with the soap molecule in the dirt, and it totally surrounds it. And this is called now a micelle. You should know that. Now, micelle is really a totally three-dimensional. In the center here is the dirt. And these little lines here are the tail of the soap, and the soap has a polar head that's the negative charge, and totally surrounds it. In the book here, they cut out a slice so you can see inside, but there wouldn't be that. And the micelle, which you should know, looks polar now. The micelle looks polar to water. Water says to the micelle, I'm polar, you're polar. I know like dissolves like. That's the important part of this puzzle. Let's go down the drain together. And they do. And you should know this. Let's go through this again. If I ask on a test, how does all right, 6 points each, or 6 points. How does soap work? Be sure to show soap structure and talk about micelles. So what do? Soap. Nonpolar tail. That's a lot of CH2 groups and a CH3. Nonpolar tail. Polar head. Water. Polar. Dirt and grease. Nonpolar. And the important part of the puzzle, like dissolves like. And if you have your dirt, The soap tails are tracked to it. The polar head stays away from the dirt. And this is a micelle. And a micelle, because the polar heads totally surround the outside, the micelle looks polar to water, and therefore, like dissolves like. Water looks at the micelle, says, I'm polar, you're polar, let's go down the drain together. And they do. And you should know all this. Go back and review this. How does soap work? Leaving my class, you should know how soap works. And later today or tomorrow, whenever you see this video and see this, after you wash your hands, while you're washing your hands, you're making micelles. And that's how you get your hands clean, your clothes clean, your dishes clean, your car clean. You use different type of soap or surfactants and laundry detergent and cleaning your hair or your hands, but it's still all a non-polar tail, polar head. Now, quick thing, a couple of years ago, a student in organic chemistry, when I taught this, came to me with a product they really liked. And that product, there are a couple companies that make it. It's called, if you can, let's make sure you see this on your screen. Yep. Like this one, Garnier or Garner, micellar cleansing water. Guess what? There's no micelles in micellar cleansing water. You form the micelles by having soap in there when you're cleaning your face, getting your makeup off or anything. My students, women, tell me these are really good products for doing that. But you have no micelles in that product. You make them when you react them with your skin and rub them with water. There's water in there too. Let's see something. Now, if we look at this product, I don't know, Garnier Skin Active Micellar, it has hexylene glycol, glycerin. This is your soap molecule and other things to make it work good. But mainly it's water, alcohols, and this is your actual soap right here. Long polar tail, non-polar tail, polar head. All right, let's move on. All right, next thing I would like you to know is how does a towel work? You know that magic device? Your hands are wet, you go like this on the towel or paper towel, and they're dry, or your face, and you wipe it off, and it's dry. How did the water get off your skin onto the towel? Is it magic? No, it's organic chemistry. Now, towel is made up of cotton, and you can see on your screen the structure of cotton. Now, do not write this down, but the important thing is see all the OH groups, that's called a hydroxyl group, bond to carbon. And those are called alcohols, not the kind you drink, but they're still alcohols. And cotton has all these alcohols, hydroxyl groups. And as I taught you with water, water has this slight negative charge, positive charge, because of the different electronegativities of hydrogen and oxygen. If I have a second water molecule, the negative oxygen, slight negative charge, attracts the slight positive hydrogen, and this is called a hydrogen bond. It's not a real chemical bond, but it's just enough force to make a huge difference. That's why water is a liquid at room temperature, not a gas. Now, if we take the same water molecule and we look at an alcohol, and this is attached to the cotton, and again, you're going to have this is slightly positive, this is slightly negative, and water can form a hydrogen bond to the alcohol on the cotton. And you should know this and be able to describe how does a towel work. And it works by how does a towel work? It hydrogen bonds cotton to water, and that's what pulls it off your hands, face, or whatever. Now, paper towel has similar structure to cotton that's called cellulose, has the same alcohols. That will hydrogen bond to water, and that's how paper towels work. Now, I worked in the industry where we made a company I made the molecule, that one of the two companies in the United States that made the molecule for fabric softeners. And after a while, if you have an older towel, oh, it's not working that well. What's wrong? Well, it happens if you're using a fabric softener, it blocks some of the alcohols from hydrogen bonding. How do you make a good old towel work better again? You wash it once or twice without using a fabric softener. You just don't. And that takes the fabric softener off the surface of the towel, the alcohols, and now they can hydrogen bond to water. By the way, the best fabric softener, most effective, is the sheets, not putting it in your washing machine. I know I have 4 or 5 patents in fabric softener chemical molecules that I invented. All right, now, switch is off. Let's talk a little about biochemistry. And that's the study of chemical substance found in things that were alive, like you, me, or plants, and how they react. Now, the functional groups that you would learn in organic chemistry for organic molecules, the chemistry, the reactions occur. This is the same for bioorganic molecules. In other words, functional group chemistry in your body is the same as what happens in a laboratory or a chemical plant. Now, switch is off, but I just thought I'd show you some molecules. Carbohydrates are polyhydroxyaldehydes, remember, switch is off, or polyhydroxy ketones. This is an aldehyde. if I can spell it right. And this is a ketone. And polyhydroxyl means many OH groups, which you see right here. Now, this is called the Fisher projection, and each intersection of a line is a carbon. And the one on the left is glucose. That's the most abundant sugar or carbohydrate. You find it in your blood. When you eat starch, you make glucose. And fructose is the sweetest sugar available in nature. And you find it in a lot of food, high fructose corn syrup. Now, table sugar is fructose and glucose bound together. And when you take sucrose, switches off here, plus acid and water, that's in your stomach, that's the HCl in your stomach. That's why the pH of your stomach is so low, because it's got hydrochloric acid. This breaks it apart to glucose plus fructose, because sucrose is glucose molecule bonded to a fructose molecule. And when you eat it and digest it, it breaks it into these that your body uses. And that's another class of how it uses it, not mine. Now, starches are polymers or glucose molecules linked together. Think of a freight train and think of boxcars. Each boxcar in a long freight train, well, those boxcars are the glucose molecules. And when you eat any kind of starch, and in your stomach you have acid, HCl, and water, and also in one of your intestines this happens, you get back a lot of glucose. And that's what your body transports in your blood to the cells that use glucose for energy. Now, I'm way beyond my knowledge of the human body. Now, proteins are polymers of what we call amino acids. And your body breaks down proteins, switches off, let's say acid, HCl, and water, to amino acids, which your body now can use to build more proteins by putting amino acids together, which your body also does. They get it from the food you eat. You build protein like your skin, your hair, and your muscles. See my muscles? Yes, Dr. White still works out with weights. Now, I talked about steroids, and steroids, again, switches off is your hormones, and they're based on this 6-membered, 5-membered rings. Remember, every bend in a line is a carbon, like here, here, and so on. And you have A, B, C, D. In chemistry with steroids, that's the A ring, B ring, C ring, and D ring. And all your hormones, which I talked about, that control things in your body, have that ring system. And with that, I'm finished with the organic chemistry chapter. Now, tomorrow, I will go through the review for test number 4. Well, I gotta get this finger fixed. Test number 4, that will be on Monday the 8th. Tomorrow, I will go through the organic problem set. And tomorrow, I'll remind you again about the extra credit project. Now, Monday, I have a special treat for you. I will be doing something, showing you something, deals with chemical plants that most people never see. I will be cutting it out of the video and not posting it anywhere else. So if you want to see something really neat about chemical plants, come Monday, the 8th, to our lecture. With that, I'm going to end a little early, because don't forget, yesterday we spent a whole lot more time. With that, I'll say, Gang gesund, goodbye.